The next on our agenda is the Lifetime Achievement Awards panel. I would like to welcome the panelists. We should have two of them joining via Zoom, Elliot Meyerowitz and Jeff Dangl, and we should have three awardees in person, Joe Ecker, Joanne, Chory, and Chris Somerville. Just stay here.
Okay, I'll just stay here. I'll be Vanna White.
While our panelists are getting settled, I'd like to explain what this award is all about. The award is named after Philip Benfey. The reason for this choice is the fact that he exemplifies exactly the traits we were looking for in a Lifetime Achievement Awardee. Outstanding research, focusing on Arabidopsis, effective mentorship, and community service. And the people that are currently in Zoom and people that are currently on stage are excellent examples of the traits of an excellent mentor and a scientist and a community serviceman. So while we are distributing microphones, I'd like to again say who our awardees are, and I will start by reading short quotes from their past trainees. Jennifer Nemhauser, a former trainee of Joanne Chory, stated, "I love being in Joanne's lab, and I'm so excited to see her honored with this wonderful award. If I was to sum up Joanne's mentoring style in 3 bullet points, they would be, don't let your ego interfere with your science, follow the data, and find the mechanism. Number 2, be brave. Try out lots of ideas, some of them kind of far-fetched. 3, surround yourself with people that love science. And remember not to take yourself too seriously. This is supposed to be fun." Jeff Dangl's quote comes from Jeff Chang, his former trainee. He notes that the characteristics that set Jeff apart are his unmatched ability to digest, synthesize, and simplify information from diverse systems, and his skills in communication, management, and mentorship. Dr. Terry Long, also former trainee of Jeff, states that throughout my career, Jeff has remained one of my closest mentors. I know that I can call upon him at any time, and he will be there to provide wise counsel in a blunt and upfront manner. His encouragement and mentorships have been hugely influential, and I truly value his dedication to my career and that of many others, especially given his amazing scientific contributions and his vision and leadership within the Arabidopsis community. The quote from Joe Ecker comes from Ryan Lister, who states, "I know many past trainees from his lab, and across the board, we consider the time we had in his lab as the best period of our scientific careers. His unbridled enthusiasm, passion, drive, and innovation infects everyone with a can-do attitude to push the limits and try new things. He builds an unparalleled research environment with excellent guidance, opportunities, training, resources, and provision of space to independently explore and experiment." And Dr. Jose Alonso notes that Ecker's innovative and visionary approaches to plant biology research have permeated the Arabidopsis community worldwide, not only through his many high-impact publications, but also by passing along his high standards for plant research to his many US and international trainees.
I'd like to read a couple quotes for Dr. Elliot Meyerowitz from his former trainee, John Bowman. John Bowman ascribes Dr. Meyerowitz's success in fostering scientific offspring to his mentoring style, where he provided ample freedom to pursue research avenues and encouraging independent thought, and yet was unselfish with his time to provide a thoughtful answer or sage advice. Dr. Karen Chang reflected that Elliot provided exceptional insight and guidance and is always available to discuss science. He is unfailingly positive, honest, and fair. He treats us as colleagues, never as underlings. He supports us in pursuing our interests. Notably, his office door was always open. His office was the hub where the lab members were welcome to hang out at all hours. There was a couch, chairs, coffee, and a coffee table piled high with journals. Elliot installed a desktop computer for the lab to use next to his own desk, so when we worked on the computer, we sat 2 feet away from him. I'd also like to congratulate Chris Somerville. Dominique Bergman describes, from the vantage point of one of Chris's postdocs, Chris struck me as someone who absolutely loved science and absolutely loved doing it efficiently and effectively. He was a great delegator. He'd teach you to do something, in my case, how to plant Arabidopsis and how to prepare a paper's narrative down to the essentials, or give you the tools to learn it from another source, and then assess whether you'd mastered the skill. If he thought you'd mastered the skill, he trusted you from that time forward. So congratulations to all our lifetime awardees.
The PACLA Philip Benfey Awards come in with a wonderful Arabidopsis-themed mug, custom-made specifically for this occasion. And the certificates.
And the certificates.
The mug is really wonderful. I drink coffee every day with it holds a lot of coffee too. It's really big.
So now we will start the panel. And the first question we will ask is briefly, when was your first encounter with Arabidopsis, and what convinced you to work on Arabidopsis as your model organism? We will, because we've got 2 on Zoom, we will have to call on people. So maybe we'll start with the Zoom. Elliot, would you like to answer that one?
No, but I will answer it. My first encounter was when I was a graduate student at Yale. I was working on Drosophila brain development, but I was very interested in plant genetics and hung out with people in Ian Sussex's lab and took the graduate seminar in plant genetics and development that Ian taught. And one of my colleagues, Jim Wong, pointed me to a review article by George Redeye about Arabidopsis, which Jim and subsequently I thought very much resembled Drosophila in its genetic capabilities. And after that, and that article was in 1975, after that, I was determined that I would apply Drosophila genetic methods to plant genetics using Arabidopsis.
Thanks. Jeff?
Well, my first encounter with Arabidopsis was when I was a postdoc in Cologne at the Max Planck, and there was a fellow there named Csaba Conch who was developing insertion mutagenesis to make loss of function mutations. And the time I was, before that, several years before that, I had been very smitten with the notion that plants must have a dedicated immune system, reading lots of papers, and thought, well, if only we could select mutants that lost resistance and somehow map the genes, then we should be able to clone them. And that influence was derived from a very influential paper published by Ray White, Ron Davis, and David Botstein that showed that you could positionally clone genes either from crosses between 2 natural variants or between a mutant and an outbred parent. Then I had met Elliot when he was a postdoc and I was a graduate student, and he was already discussing using Arabidopsis as a genetic model. So I believe in 1985, 86, I guess, we went from Germany back to California to visit my family, and I invited myself to horn in on Elliot's holiday, and he was kind enough to give us all the RFLP markers that existed at the time and the rudimentary RFLP map that he had made. I should also note that when we were in graduate school in the early 80s, we read Stephanie Wittweiler's famous paper on the small size of the Arabidopsis genome. So those things in combination convinced me that it would be a great model to look for the plant's immune system.
What a fascinating story. So we're going to move on to Joe, then Joanne, then Chris.
My first encounter with Arabidopsis was when I was a postdoc in Ron Davis's lab. And coming from the animal field, when I started in the lab, I wanted a system you could do cell culture. And so I started out actually looking at somatic embryogenesis in carrots. And then, you know, we did a lot of experiments, transit, transfections, et cetera. But then I said, we need to do genetics. So then there's a fellow, I can't remember his first name, his last name was Simons. And he was doing genetics in carrots, which required the use of meat flies in a giant bag, you know, covering each one of the plants. And I thought, you know, this is not a great genetic system. And so Chris was kind enough to send seeds to Ron's lab, and we had seen Leslie's paper on the small size of the genome. And Ron, I think, had come down to Caltech to give a talk and talk with Elliot. And Chris sent seeds to Nigel Crawford, who started working on nitrate reductase, and me being across the bench from Nigel, got some seeds and then started in that way. So it was the influence of at least two of the people in this session that got me started.
All right. Joanne?
I don't have a microphone.
You have a microphone on your shirt, so you should be good.
It's just the one on your shirt.
Oh, that's good. All right, sorry. Thank you. I still need these guys to do anything. Anyways, I remember quite well the first time I thought I would work on Arabidopsis. It was my first year as a postdoc in Fred Oswald's lab, and Fred had gone to a Gordon conference where Chris had given apparently a stellar talk. So Fred went out to the local lake, you know, these mountainous clear-water lakes, and he was windsurfing, and he got banged on the head. This is what he told us, anyway. You know, so we weren't sure if it was the concussion talk or Fred talking, right? But anyways, he got banged on the head, and he woke up and had an epiphany. And he said, we're working on Arabidopsis. Go get Joanne and bring her down to my phone. And Neil Shevsky and I were instructed by Fred where to find the seeds that he had gotten from, I think it was George, Red Eye. And so anyways, that's my story.
Really cool. Chris?
I did my PhD in E. coli, and after my PhD, I rented an apartment in Paris to think about what to do with life. And Shauna, my wife, and I spent our mornings in the Institut Pierre and Marie Curie thinking about what to do. And we decided that she had done a degree in plant breeding, and so we were painfully aware how backward plant breeding was. And at the same time, just before I graduated, I'd gotten a gift of EcoR1 from Howard Goodman, which you couldn't buy yet. And so we could see that cloning was going to come. And so we started looking for a plant that we could do modern biology on, basically. And we ran across George Redeye's paper, that Elliot 75 paper that Elliot mentioned, and decided we would try and create a group around it. Right from the beginning, we had this idea that we needed if we're going to make a model organism, we needed a group around it, and we started thinking about how to do that, basically, right from the beginning. That was 1978. Yeah.
Thank you. What fun stories. The next question is, what is one thing you felt the most excited about or proud of during your career? And I know there's many, but keep it down to like one brief one. So we'll start with people here. So Joe, do you want to start?
It's a tough one. I was excited about a lot of things. You know, when somebody, I think, develops something new in my laboratory, some kind of new method particularly, that gets me excited. So like was mentioned by Jose, the DAPSeq method. Suppose Ronan O'Malley comes in my office and says, hey, I'm going to do this assay, you know, DNA affinity purification. And I said, yeah, maybe. And he goes off and then comes back with some data. I said, wow, or maybe he had it already and said, yeah, if I would have said no, he would have showed me up. But when I was a postdoc with Ron Davis, and this is a message that, like, say, if you're building a community, like Chris said, you need to spend some effort in developing reagents that enable everybody. Otherwise, there is no community. You're always stuck. Jerry Fink used to give a model where you'd have the pillars of a house, and it was, you know, Arabidopsis was always sitting on like 2 pillars or 3 pillars, but never 4 pillars, not really stabilized because we didn't have the tools that we needed. So I get excited most about, you know, people developing new things.
Excellent. Joanne?
Okay. I think the moments I was most proud of in my lab were when we cloned a gene for a receptor, say, but not only just identified a candidate for the gene, we actually went on and did the work that showed it was really a receptor. You know, so receptors, as you guys know, they bind some substance or protein, and that causes a biochemical change in the receptor itself. And so when Toshi came to my lab and showed that BRI1 could bind steroids, he showed it was direct. And that was an important thing that we showed. And we also showed it in other ways with Jian Wang. So, I mean, that's an example of one. Finally, we brought that receptor down to a crystal structure. And again, we did this with colleagues, Ian Wilson, who lived right up the street from us, and Jonah Well. Yeah, so I think those moments were the best ones for me because it showed we could go from the genetics all the way to the mechanism. And believe me, I didn't sleep well for like 13 years, waiting for BRI1 not to bind directly to Brucellite, right? Because I had proposed that model, and that's what everybody always remembers. You show that thing as a steroid receptor yet? Nope. I'm going back home right now, you know. But anyways, it was kind of an interesting time for me, but I was really happy when we cloned these things. So, I guess that's it.
Sounds exciting. Chris?
Well, I guess what makes me really happy is the success of the community, because that was something actually we all, I think, wanted. But my best day, I think, is that at some point we, as a group of us, actually decided we needed to raise the money to sequence Arabidopsis. And Elliot and I, and maybe others here, used to teach at Cold Spring Harbor in the summertime. And Jim Watson used to come around to the cafeteria in the mornings and see who was there. And so I knew, and Elliot knew Watson. And so we invited him to come to a meeting with the director of the National Science Foundation with us, and several others. I think Ron Davis, I think, came, and I think Ollie Nelson, who was a well-known maize geneticist, came with us as well. And basically we got there, and Watson said, oh, these young guys have good ideas, you should listen to them. And that was his contribution. And then we asked for $100 million to sequence Arabidopsis. And this guy was the director, I don't remember his name now, but he was so excited by it, he called in the head of the biology division and said, we're going to do this Arabidopsis project. So he basically agreed to give us the $100 million that day. So that was definitely my best day. And on the way back to the hotel, just as a matter of interest, I was walking back with Watson, and he said, oh, this meeting confirms one of my life experiences, that sometimes it's easier to get $100 million than $1 million, because when you ask for $1 million, they ask you for a 25-page proposal. But when you ask for $100 million, they just ask for a concept. I don't know, maybe Elliot remembers it differently, but he was there too, so anyway.
What a story. Elliot.
Oh, the thing I'm most proud of is the success of the students and postdocs who went through my lab. And I'm going to share my screen if you'll let me. Can you see this?
It's coming. Just a second.
Not yet.
There we go.
There we are.
Just as an example, Adrian sent me this picture from the Cannes Convention Center at the International Plant Molecular Biology Conference of the gathering of former members of my lab who were there. And I'm just so proud of the successes all of them and all of the others have had. I mean, for me to get this picture made me much happier than any paper I've ever published.
Thank you. It was a fun time. Jeff.
Well, I'm going to go with Elliot. The proudest thing of my career is the 75 postdocs and 34 PhD students who nearly all remained in science and nearly all remained in touch. So Sarah and I are super proud of that. I think my most exciting moment in Arabidopsis research, I guess there's two. The first was when I did a cross between Columbia and Landsberg, and in the F1, the erected phenotype was lost, and I knew it wasn't a self-pollination event. And for a mouse immunologist to successfully cross two plants, that was a big day for me in the lab. And the second most exciting thing was some of you in the plant microbe field might know about a protein called RIN4 that we studied extensively for a long time. That was actually cloned by an undergrad in the lab. And when he came into my office with the sequence, I thought he was so excited. He was definitely not touching the ground. That was a really great day too.
Great story. We're going to move to the next question, and we're going to start with people in Zoom. All scientists experience some ups and downs in their careers. Do you recall any moments in your professional life where you doubted your ability to keep going? Jeff, do you want to start?
Well, the flippant answer would be pretty much every day for the last 40 years. And there's some truth to that. The answer is, I learned this from a postdoc that I worked with as a graduate student, Vernon Oy. And Vernon said, don't take the highs too seriously, and the lows won't bother you. And so I've tried to live by that as well. Of course, there's ups and downs. That's science. Projects crash and burn, and you have to live with that. I think it's actually more important to learn when to kill a project than it is to decide what to do next in a project that's going well.
Thank you for sharing your wisdom. Elliot.
I'm not really sure how to answer this because I've never felt that I had a career or a professional life. I've always felt that I'm just having fun and amazingly getting paid for it. And so I just never had the mindset that anything that happened to me would bother me that much. And so I don't recall any moment when I doubted my ability to keep going, but not in a profession or a career, just get up in the morning and go do what I love.
This is a great attitude. Thank you. Chris?
Well, I also don't remember any such occasion. However, I did stop working in plants about 10 years ago for all kinds of reasons. The main reason, I think, was that when I was at Stanford, I inherited a group of ecologists, and they got me all bent out of shape about climate change. And then next thing I knew, I was leading a big energy center at Berkeley with mostly chemical engineers and chemists, and I got further and further away from plant biology. So when the price of oil crashed in 2015, I decided to leave that and now I work for one of the Facebook founders, actually. I give away his money for biomedical R&D. And, you know, I miss plant biology, but I feel like I'm on my third PhD because I've given away more than $500 million in the last few years to support biomedical R&D in human medicine, and I found it very, very interesting to learn that field, basically.
That's a great story. Joanne?
I think the worst day of my life was the day I took the first year oral exams. I blew them, you know, and I handed it in to this professor named Ralph Wolf. The guy's a great microbiologist. He looked at one I wrote down from one of the answers, and he went like this. Oh, God. And so anyways, I actually applied for a job at Nova. I don't know why I thought Nova was the answer for me. They didn't call me back, so I stayed in graduate school. And I didn't have another day that was worse than that one. And that was good. So anyway, it's just a silly little story, but...
So the take home that anybody can bounce back. That's great. Joe?
I don't actually recall any time when I doubted that I would continue, but continue in one area. I have changed, as Chris said. I had to get out of the field of virology because it didn't have it had a very nasty community. It was super competitive. Nobody talked to one another. And then the idea of kind of transitioning into a community like that was being built in Arabidopsis was very attractive. So I haven't had any experience of wanting to give up, but I have given up fields over the years. And even now I'm sort of transitioning into another area, like Chris said. And it's exciting. You have to keep changing, I think. Changing institutions. Joanne, she didn't have to twist my arm too hard to move, you know, to the Salk. But I think change is a good thing. And it keeps you motivated. Then you don't have to say you're doubting yourself. You'll find something new.
Excellent. Our next question is, what big questions in plant biology are still best addressed using Arabidopsis? And we can start maybe with Chris.
Yeah, I think there's still quite a bit of dark genome in Arabidopsis, and I think that we should try to get to the root of what all the genes are doing in at least one organism. And I thought this morning's talks were really great, actually, by the way, and good examples of dark matter revealing itself. You know, Sue's protein is a really novel and interesting thing, and also Jose's the novel proteins I thought were good examples of new functions, and I think there's a lot more there. The question is how to really how to assign function to those genes, I think. Having said that, I want to say one more thing, which is I'm still bent out of shape about climate change, and I do think we should be really thinking about how to develop plants that, you know, are going to maintain productivity, because population's still growing, and I think the amount of arable land is going to change at the very least. If it may diminish or it may change, but I think for example, in North America, I can see crops moving north, but in order to do that, we're going to have to change the photoperiod of most of them. And I don't think that work is very robust right now. And so, but we know that from Arabidopsis, at least, we know what the genes are that should be involved in that. And so there's a big opportunity right there, I think, to start. But I could go on, but anyway, I'll stop. Thank you. Joanne?
I still think Arabidopsis is the organism extraordinaire for doing genes discovery. And so anyways, I'm saying Arabidopsis has to stay in the picture, but it might also help if people just did some more hardcore biochemistry, which I think a lot of us don't like to do. So anyways, what was the question again?
What big questions in plant biology are still best addressed using Arabidopsis?
Okay, so another big question in plant biology where we need to have more input is how all these pathways interact within a cell. And I find it really kind of confusing, you know. Plants have so much redundancy built in, but then they take the plant hormones and they make them all do the job, or, you know, they don't compensate for each other. What's going on there in minds of those plants? And I also think we need to show, and I think we can show it in Arabidopsis, how a brain works when you don't have a structure, like a big, complicated structure in the brain that Joe Ecker works on. I think maybe the epidermal layer is the brain for growth, because I think that's where a lot of these pathways actually come together. And so to do those experiments is a lot of work. You have to be able to separate cell types out, and then you have to be able to show they're molecules talking to each other in there. And so, yeah, I guess that's what I would say.
Yeah, intersecting pathways, one of the big mysteries. Excellent. Joe?
I think pretty much everything is on the table, right? So we hear about new, like Sue's example of, you know, disorganized protein and what it does. You know, that takes a lot of work. So there's sort of the mechanistic aspect, but there's also the evolutionary aspect, how like Moise talked about adaptation. You know, we need to know where things are happening within cells, sort of working your way out, what are the cells, how they communicate with one another. So I think there's a future for all of you. I really do. I think that, you know, if we can keep Arabidopsis on the forefront, then, you know, it's great. Other people that are working on crop species, or if your labs work on both, fantastic. But I think it still has to be a leading edge organism, and I think it still is. I think it'll be for quite some time. CRISPR doesn't do everything. It does a lot, but there's a lot of other things that you can't easily study in an organism other than Arabidopsis. So I think there's a big future for all of these mechanistic studies all the way through evolution.
Excellent. Jeff?
Well, I'm going to agree with Joe. I think everything in plant biology is answered best by the system in which the best tools are available. And that certainly is Arabidopsis. The challenge will be to keep funders interested in Arabidopsis. So my suggestion for the big questions would be, and Jose talked about this during his talk, how do you use Arabidopsis, genetics, genomics, and ecology and evolution to answer important questions that are comparative in nature? So how do we make Arabidopsis the premier model plant to study all plants? And I mean that in a way that's a bit different than it's been in the last 30 years. How do we make Arabidopsis the key tool that you rely on when you want to step out into a different system to understand how that system may have evolved?
Thank you. Elliot?
Well, I think all the big questions in plant biology are still best answered using Arabidopsis. If they're not best answered using Arabidopsis, maybe they're not such big questions. The real discussion here is, what are the big questions in plant biology? And I think with Arabidopsis, we've developed methods to deal with genomes and genes and gene expression that are really world leading and equal to that in any organism, plant, animal, or fungus. But there are other things going on in plants. Joanne pointed out biochemistry, and I would point out fundamental cell biology, where I think we have to develop Arabidopsis to answer questions at those levels so that it continues to be the most important plant for answering big questions in plant biology. And so I think we've got some work to do in developing other community capabilities for Arabidopsis beyond the ones involving genes and genomes, including the ones involving proteins, but onto basic vesicular traffic, other sorts of nuclear structure, other sorts of basic cell biology questions, where I think Arabidopsis could also lead the world, and we just have to develop it.
Awesome. The take-home message, we're going to stay in business for a long time. At this point, we would like to start taking questions from the audience. Please come up to the microphone, and you can ask a question directed to a specific panelist or a question to all of them.
Don't be afraid. We won't bite.
Okay, while people are warming up, I'm going to have a list of questions for you to think about that you may want to ask. It's like guidelines. If any of those questions speaks to you, please feel free to come up. I think Jiyoung already has some question. Go ahead.
I would like to ask your view about the future of plant biology, and a particular point is how you think AI will change the game.
Great question.
Well, yesterday there was an email that arrived in several of our mailboxes that are HHMI supported that said that they're going to build a $500 million operation that you'll be able to at least access, I think, through the community, $50 million a year for 10 years to develop AI methods at Genelia that people can use. So I think it's not cheap to do this. You need organizations like that to step up. You know, there's going to be a similar initiative happening at CZI. Steve Quake's operation is going to do that. I think there's a lot of opportunity to build onto some of those models. Many of them will be basic sort of gene regulation that can also be inputted with smaller transformer models for Arabidopsis to make predictions about phenotypes and things. So I think there's going to be, you know, I hope that plant biology will be part of it and not left behind. There are lots of companies in the medical sciences that have already been doing this at a very high level, but it takes, you know, $100 million to build you know, sort of a large language model that built with data. And so I hope that plant biologists can participate in that through maybe some NSF initiative that involves their division of engineering or computer science. I think it would be fantastic.
Any of the panelists want to continue the discussion of the future of plant biology?
Well, I would say some things about AI. Two opposite things to say about it. One, it's clearly going to be extremely important for us if you just look at AlphaFold, where now you can look up any protein you want and get a likely structure for it without experiments based on comparisons and first principles. And so that and querying databases, I think, is going to be a lot easier to get more meaningful answers about what's in the literature and about what any particular protein or gene might be doing, even better than excellent databases like TARE. And so I think they're going to facilitate our research in really important ways. At the same time, I've looked up a number of things using ChatGPT, and I ask it questions about plant biology, and what I get is a 7th grade essay that's disappointing at every level, even though it always has a topic sentence and a final paragraph, just like your 7th grade teacher told you. And so that on the one hand, I can see it's going to be tremendously valuable. On the other hand, it doesn't look like it's going to replace us yet.
Excellent. Okay, more questions from the audience?
So, Chris, you said you feel like you've done 3 PhDs. So this is for everybody here, except Chris. For you, like, what would you do if you had a chance to do a 4th PhD? What would that be? And for the rest of you, if you had a chance to do another PhD, what would that be today?
My answer would be I'd be a traitor to the group if I answered that honestly. I mean, you know, the thing that I'm most curious about actually is what the hell is consciousness. I can't understand it. Everything I know about cell and molecular biology, I still can't understand what consciousness is, even in concept.
It's in the colostrum.
So
That's what Francis Crick always said.
If I had more time left, I would go into trying to understand that, I think. But, yeah. What do you guys?
Well, I kind of like how organisms talk through chemicals to each other. That would be a field I would find very interesting to be in. And I don't know what else.
I'm happy with what I'm doing. I don't want another PhD.
You're working on my problem, aren't you?
Jeff?
Well, I was thinking, I worked on mice and humans a little bit in immunology, and I worked on plant immunology. I'm still fascinated by how recognition and response systems evolve and how recognition of non-self works across evolution. So I might do something crazy like go work on lampreys, which have a fascinating immune system. It seems a little bit trivial, but I think the other major thing that I've gotten a bit interested in, Chris mentioned this earlier, I would really like to understand whether we can make ecology reductionist science, that actually you can answer questions, test hypotheses, and solve problems specifically with respect to climate change.
Interesting combination. Elliot?
Well, let me say two things. One is if I had my undergraduate to do again, I'd take more math classes because I'm struggling at my age to catch up on vector calculus right now. And so that would be my first recommendation. But as far as new areas of research, there are a couple of really great frontiers I'm finding fascinating now, both of which relate to plant biology. One is algology and what sort of algae live in the oceans, and the diversity and the strangeness of some of those organisms is extraordinary, and they're so little studied. So I think there's a huge frontier there for intellectual adventure. And the other direction is into the soil. I think soil is the most complicated ecosystem we know about, and to find out what's happening in 1 cubic centimeter of soil would be a lifetime's work, and it'd just be so much fun.
Fun. Okay, more questions from the audience.
Thank you very much. I'm very happy to be here and have you guys. It's very great and great example for the new generation. So obviously I'm young in my career and I have a few supervisors. Some were in the early career, some were in late career, and the one in late career, they had a favorite sentence, which I think they all have. They says, back in the day, and they were always saying like, yeah, back in the day was like that, and I found in like they were missing something. So what is your back in the day that you are missing today?
Back in the day, when you sent a manuscript in and it would come back accepted, no changes, okay? I haven't seen that in decades, but it does happen. I can give you a letter like that shows it. It's like, wow, okay, that doesn't happen.
Joanne, what is your back in the day story? Back in the day, what do you miss?
Back in the day, if you went with if you thought a paper was more or less sound, you didn't sit there and grab over every little detail. You know, I mean, I would like to see us go back to authors taking responsibility for what they put in their papers. And if the reviewers kind of buy the idea and think enough data support, then I think it would be good to just accept the paper. Maybe that's what's going to happen with these servers. You think? I don't know. But yeah, back in the day, postdocs didn't write all the reviews. Maybe I should say that. Anyways, yeah.
Was it easy to find reviewers back in the day if you were an editor of a journal?
I always found reviewers when I did the genetics editorial board. We had to find our own reviewers. They had no centralized office back then. I always could find reviewers for that. I did the journals found reviewers that were pretty relevant. I don't know. They could find reviewers, yeah.
It became excruciatingly hard to find qualified reviewers that are willing to review. So
Yeah, that's right.
I guess it was a little bit easier back then. Chris, what is your back in my day story?
I don't have anything in the past that I long for. I love the ways where science is now and the capabilities that we have. It's just I'm amazed by it every day now. And mostly my thoughts of the past are, well, wow, that was so primitive where we were, compared to what's possible today. And so I guess the only thing I wish for in the past is my youth, so I could use all the tools of the moment in science. And
So you no longer miss making your own ICAR 1, right? You can
Nope.
just place an order
Don't
and
miss any of that
overnight it's
stuff.
shipped to your
Yeah.
door. Jeff, do you have any back in your day stories?
I think back in the day, it took us 1,000 times longer to get anything done than it does today. So I have no qualms about leaving back in the day.
Really cool. Elliot?
Well, I'm with Jeff. I don't miss anything. The only thing I miss about this field and my work decades ago are some of the amazing personalities who passed on. I mean, there's some remarkable old scientists I admired then and I admire now, and I miss them.
That's a good point. More questions from the audience.
So let's say you are in a room with a bunch of philanthropists who are so eager to fund basic medical research and cancer biology, and you are there to persuade them to send some money in Arabidopsis research. Not 100 million, but just a small amount of money. So what would you persuade them in a few words, a few sentences, like for those to attract some money in the cancer biology research into Arabidopsis system?
I'll take that one.
Go
I'll
ahead.
take that one, and I'll ask Elliot to please give us his famous quote about the squirrel.
I don't even remember it.
The quote is from the interviews that were done by NPR radio after the genome was sequenced. And the question to all of us was exactly this: why should we care about plants? And my answer, being an immunologist, is plant health is human health, because without plants, humans would be in deep trouble. Elliot's answer was, he was looking out the window of his office at Caltech, and he saw a squirrel, and he realized that everything the squirrel ate was a plant, and everything that ate the squirrel was therefore eating the plant. And so we all require plants. And I think people actually, it's been my experience with funding agencies and the general public, that if you tell them and point out to them the completely obvious fact that we rely on plants for all of our energy, and they say, oh, wow, I never thought about it that way. And they're more receptive to the discussion. In plant immunology and plant disease, there are now currently at least 5 major epidemics going on in the world that are killing, threatening to wipe out crops that we eat and enjoy all the time. So I think a compelling argument is quite easy to make for why we should care about plants.
I would say it depends on who I'm talking to. Who's the philanthropist? If it's Elon Musk, I'll say, hey, we've got this great program to put plants in space, right? And I think a few of my postdocs are working on that with NASA, because you're going to need plants up there. So thinking, if you was talking to Mark Zuckerberg or talking to the folks that started the Altos labs, I'd say, you could use plants to study processes like aging, right? And so I think there's a lot of opportunities. I think, talking to Joanne gets a call. Will you take a call from Jeff Bezos? Yes, I will. And that ends up in, I don't know, how many millions of dollars? And that's to put carbon into the ground, right? So there's a lot of opportunities to sell what Arabidopsis can be useful for studying. And we just need to get the ear of philanthropists like his bosses. They have, every philanthropy has their own ideas about what's important, and you just have to make the right connection.
Yeah, when I went to philanthropy, I thought I was going to be able to liberate money for plant biology, you know, but there, it turns out the org I work for in has a different model. It's entirely based on what are called DALYs, disability adjusted life years, and so I've not been able to make the case as yet that, you know, the money should go towards plants rather than, you know, another vaccine for the developing world or something, unfortunately. But there's a lot of billionaires in Silicon Valley, and we at the philanthropy I work for, it's called Open Philanthropy. By the way, we were the co-founders of OpenAI, just as a matter of interest. And one of our biggest annual expenditures is in AI safety, actually. I know of billionaires in Silicon Valley that are interested in plants, and so I do think there is an opportunity for you know, some spokespeople of plant biology to get organized and go after philanthropic money, and it's there in vast amounts actually.
Is there anyone else who wants to volunteer an answer? Okay, I think we're good. Thanks for the great insights. We are out of time for the questions, but I want to again congratulate our Lifetime Achievement PACLA awardees, and thank you for all of your wonderful insights into your experience and the future of Arabidopsis biology.
Thank you.
It's very rewarding to see you all still pursuing Arabidopsis and building it and making discoveries. Actually, you know, this is what I think the founding groups really hoped for, and so it's very rewarding. That's all I can really say, I guess. Yeah.
Look at us, we made it. 300 talks over the last several days, and we're still here and we're still alive. Thank you so much for staying till the very end. To close off, I have a couple announcements to make. The first, and this is a really nice segue from the panel we just had, several of the people mentioned Red-Eye, George Red-Eye. And so the ABRC, the Arabidopsis Biological Resource Center at Ohio State, has the George Red-Eye Award. And the George Red-Eye Award, George Red-Eye, just if you don't know, came to this country and brought Arabidopsis to North America. He came to University of Missouri, which is in Columbia, Missouri, which is why the stock is called Columbia. It's not from Columbia, he didn't take it out of the ground in Columbia, but he named it for that city. And so it's very important that we contribute our stocks to the ABRC, and I encourage you to contribute stocks. And every year, the ABRC gives an award to the person who donated the most stocks. And today, we'd like to thank Dr. Diana Wolf from University of Alaska Fairbanks. She donated 92 lines of Arabidopsis kamchatka, which she had collected and now are available at the ABRC. So as an award, she gets 75 free stocks from the ABRC. So let's give her a round of applause. Okay, so then I have two announcements on the dorms. One, if you're checking out of the dorms and leaving today, you can't just walk away from the dorms like you would at maybe a hotel. You have to actually check out and turn your key back in. Second, obviously there's a lot of travel delays today. And if you're having a canceled flight or travel delays, there is a potential to continue to stay in the dorms. But you need to please contact Joanna Friesner right there by at least 3:30 today. They need numbers by 3:30. So you have an hour or so, but you can't wait till late tonight. The third announcement I have is please look for an upcoming post-ICAR survey to the community that helps direct us on what went well, what didn't go well, and what we need to change. The fourth is I'd like to invite to the stage now Dr. Daniel Van Dam from the Ghent University. Daniel here is the host of ICAR 2025 in Ghent, and I think we're all really looking forward already, starting now, to next year. All yours. Well, first, I'd just like to publicly thank a number of individuals here. So maybe now would be a good time while they get this sorted to have all of NAASC come up on stage. I'd appreciate that. I think you guys are seated mostly in here, and we'll try to steal Joanna as well. Can we get you on stage here, Joanna? Can we get you on stage here as NAASC? So obviously we need to thank the UCSD. The staff has been terrific, and I really appreciate what their efforts they've put into this. The Salk for their generous donation and hosting us for the terrific party yesterday. Thank you, the community. You guys have been terrific. You guys have been super collaborative, super energetic, and I really appreciate, especially you guys sticking around to the very, very bitter end. I need to thank these guys, NAASC, for all that they've done. They've been working behind the scenes tirelessly to put this thing on. Lots of, we use the duck analogy. We look, it's hopefully calm above the water, but below, it's been chaos, right? So thank you, NAASC, and particularly thank you, Joanna, that you've made this possible, and none of this would have happened. We got you a gift. And we hope you enjoy it. When you Zoom with Joanna, as many of us often do, you're treated to like a loft where she lives in the woods, and so that's what inspired the gift.
Is that a coffee bag?
No, it's not. I've had a terrific time being the president of NAASC. Today marks the end of that presidency, and Adrienne Roeder from Cornell is the future president, and she will do a great job. I got her this. I couldn't. We got the slides. My last announcement, and then Daniel can close us out, is that lunches are here. They're, you can either eat here in the courtyard or take them to go. And thank you all so much for being here and being so collaborative and great energy.